Hello friends, welcome to Bioassembly. In this session, I will discuss about cell cycle and cell division. The cell cycle is the sequence of events occurring in an ordered fashion which result in the cell growth and cell division. That implies cell cycle is a sequence of events and it occur in ordered fashion. And cell cycle consists of two basic phases and state or periods. That is called growing phase or I phase or short dividing phase or M phase. The cell cycle begins at the end of each nuclear division and ends with the beginning of the next. The cell cycle acts as a unit of biological time that defines the life history of the cell. That implies it is a unit of biological time and it defines life history of cell. The cell cycle is continuous process that include all significant event of the cell ranging from duplication of DNA, cell organelles, subsequent partitioning of cytoplasm. In cell cycle process, uh, there are duplication of DNA and cell organelles and partitioning of cytoplasm. These are the cell cycle events. These are the I phase or these are the I phase or intermediate phase, and these are the metaphase. You can see these are the structure of the cell cycle. First of all, G, in I phase there are G1 phase or growth phase, where first growth phase where the all nucleotides and amino acids and RNA and protein synthesis and growth of nucleus. In G1 phase, nucleotide, amino acid and RNA and protein synthesis occur and also growth of nucleus take place. S phase or synthesis phase. You can see that these are the large S phase or synthesis phase. In this phase, DNA duplication, histone synthesis, growth of nucleus and replication of chromosomes, centriole replication. In this phase, S phase or synthesis phase is where DNA duplicate and histone proteins are synthesis and replication of chromosome occur. Second is a growth phase. Growth phase RNA and protein synthesis of formation of macromolecule. In growth phase, RNA and protein synthesis occur and also formation of macromolecule. These are the M phase or metaphase is also called metaphase. In M phase, there are five types of phases: prophase, metaphase, anaphase, telophase, and cytokinesis. Let's understand one by one. In addition of the process of cell growth, where the cell absorbs nutrients and prepare its cell division as a part of cell cycle. When cell growth occurs, cell absorb nutrients and it prepare for division. The process of cell cycle occur in various phases and which are specialized for particular stage or cell. You can see the structure of the cell cycle phases, various phase, G1 phase, S phase or G2 phase and M phase, metaphase. The overall process and step of the cell cycle might differ in eukaryote and prokaryotic organism as a result of difference in their complexity. You can see the structure of eukaryotic cell and prokaryotic cell, and their order of the cell cycle and cell division are totally different. So there the structure is complexity is different from eukaryote to prokaryote. These are the three main types of cell cycle: chromosome cycle, cytoplasmic cycle, and centriosome cycle. In the chromosome cycle, DNA involved in the synthesis and that alternate with mitosis. And during this cycle, chromosome cycle, double helical DNA of the cell replicate to form two identical daughter DNA molecule. During chromosome cycle, double helical DNA replicate and it form two daughter DNA molecule. And this process chromosome cycle followed by mitosis to separate the cell into two daughter cell. Second one is the cytoplasmic cycle. It involves the cell growth and that alternate with cytokinesis. During growth, cell accumulate nutrients and growth factor and double the contents of the cytoplasm. In cytoplasmic cycle, so the name suggests cytoplasm, it implies that cell accumulate nutrients and growth factors, and it double the content of cytoplasm. Eventually, the cytoplasm divide via cytokinesis equally by cytoplasmic content into two cells. These are the phases of cell cycle. You can see there the structure. First of all, G0 phase or gap phase. In GO phase is a phase of cell cycle, is a period of time where the cell present in the quiescent stage or resting phase. You can see that the structure in this phase cell are present in the resting phase, neither divides or nor grow. In G0 phase cell neither divides or nor grow. The G0 phase can consider either an extended G1 phase or separate phase out of the cell cycle. You can see that the structure of the G0 phase. Here is the G0 phase. You can see these are the G0 phase. This the small portion is the G0 phase, or is also called where cell are resting phase. And usually cell enters the G0 phase when reach maturity, like the case of muscle cell and nerve cell. When G0 phase occur, the cell when mature, like muscle cell and nerve cell, but cell continue to form either function of the throughout their life. Next one is the G1 phase or gap 1 phase. The G1 phase of the cell cycle is a part of interphase where the cell begin to prepare for cell division. A cell enter the G1 phase after the M phase of the previous cycle, and thus termed as a first gap phase of the first growth phase. In this process, G1 phase is after M phase, cell enter G1 phase, and here the first gap phase is also called G1 phase is also called first gap phase or first growth phase. In this phase, no DNA synthesis takes place and RNA synthesis occur, and in order to produce protein required for the proper cell growth, in G1 phase, transcription of all three types of RNA, which then undergo translation into the protein and enzyme necessary for the event of cell cycle. That implies in G1 phase transcription of all three types of RNA occur, and it undergo proteins and enzyme synthesis necessary for the cell cycle. It takes 50% of total cell cycle time. G1 phase is a larger phase, and it takes 50% of the cell cycle time. An important of G1 phase is a G1S site checkpoint that determine if the cell is ready to enough proceed into the division phase. At this point, event like the detection of DNA and damage of nutrients concentration occur. When, in this process, G1 phase is also called check 1 or S phase. In this process, cell double check the process ready to enough proceed for cell division or not. If cell is not ready, then it rests phase. Next is the synthesis phase. The S phase or synthesis phase of the cell cycle is a part of interphase where important event of the DNA replication and formation of histones protein takes place. You can say the name suggests S phase or synthesis phase. Here the synthesis of all important materials are occur. In this process, DNA histones protein are synthesis DNA and like for DNA replication. The process of S phase is tightly regulated as the synthesis of protein and replication of DNA require almost precision. The production of histone protein and other proteins are crucial in this phase. Here, S phase is a production of histone protein and other protein is important for this phase, and newly replicated DNA molecule required histone protein to form nucleosome. That implies that newly replicated DNA molecule required histone protein from nucleosome, and the enter into the S phase. It regulates G1S checkpoint that allow the cell to enough nutrient healthy DNA to enter the next phase, and it takes 30% of time. Synthesis phase take 30% of time, and it it has a G1 as checkpoint, and that see they allow the cell to enough nutrients or healthy DNA enter to the next phase. If healthy DNA is not not, then it is stop where then it is stop here, and no division occur. During this phase, the DNA content double and the cell, but the number of chromosomes remains same. Synthesis phase, the DNA content double, and in order to preserve the epigenetic information, different region, the DNA are replicate at different time. Similarly, actively expressed gene tend to replicate during the first half of S phase, whereas inactive genes or structural protein tend to replicate during the later half. First of all, actively replicate genes are first replicate in S phase, and whereas inactive genes are or structural DNA replicate in later half. Therefore, end of S phase, each chromosome of the cell has double the amount of DNA and which double the set of genes. Next come to the G2 phase. G2 phase of the cell cycle, where the cell collect in nutrients and release into the release protein in order to prepare the cell for M phase. In G2 phase, cell collect nutrient and it release proteins for prepare cell for M phase. The G2 phase is also part of the interphase when when the cell is still in the resting phase while the preparing for cell division. Interphase consists of three parts: G1 phase, S phase, G2 phase, and G 0 phase, where cell rest in G 0 phase cell are in resting position. G2 phase is also important as it check the DNA damage. In G2 phase, it check DNA damage during replication. When replication occur, if any type of DNA damage, then stop further synthesis to ensure the cell to proper condition undergo division, and it also check to and ensure that it proper condition and it start for further division. The phase might be skipped in some rapidly dividing cell and directly enter into the mitotic phase of after DNA replication. And G2 phase is absent in rapidly dividing cell, and in rapidly dividing cell enter into mitotic phase after DNA replication. These are the M phase or metaphase. First of all, mitotic phase. The M phase or mitotic phase of the cell cycle is the first crucial dramatic phase of the entire cycle where cell divide to form identical daughter. In M phase is a crucial step of the cell cycle where the cell divide and identical daughter. The most important event of this karyokinesis or nuclear division. First of all, M phase consists of two type of division: karyokinesis, where nuclear is divided, and second one is the cytokinesis. In karyokinesis, where the chromosomes separate into two distinct cells. First of all, the process of mitosis might differ from one organism to another, and even from one cell to another. In mitosis, one organism is differ from another organism, and mitosis begins the condensation of chromosome, and which then separate into the move toward the opposite pole. First of all, you can see these are the interphase process where the cell prepare for the division. Here the all important material are synthesis and growth of amino acid occur and the histone protein. Here's our early prophase where the you can see the ancestor, and these are the centromeres double, and the middle prophase where the all the chromosome are formed, and these are start to division. In the middle prophase, chromosome are formed and nuclear membrane are disappear. You can see these are the nuclear membrane, and this is now disappear. Here the nuclear membrane is complete, but in the middle prophase nuclear membrane disappear. You can see these are the structure of the late prophase. You can see in the late prophase all chromosome are attached to nuclear membrane is completely disappear, and all chromosome are attached to spindle fiber. You can see there the structure of spindle fiber, and these all chromosome are attached. In the metaphase, chromosome in the equational plate. Chromosome arranged in equational plate, and In the early metaphase, chromosome fiber, an intrazonal fiber and continuous fiber formed. You can see these are the chromosomal fiber and these are the chromosome are separate. You can see these are the intrazonal fiber where chromosome are separate and the continuous fiber. This you see the continuous fiber and the late anaphase where the all chromosome go to their pole. You can see these are the chromosome come to their pole and here completely complete the process of chromosome. You can see these are the attached to the spindle fiber and come to the pole and the cytoplasmic constrict. You can see these are the cytoplasm constrict and here the completely constrict the cytoplasm and spindle disappear. Spindle fiber, these are the spindle fiber and here these are the spindle fiber disappear and new nuclear membrane is formed. You can see these are the nuclear membrane is formed and ancestral ray are disappear. These are the ancestral ray and these are disappear in the late telophase. In the cytokinesis where the cell divides, you can see these are the daughter cell, two daughter cell. Here the this come to this phase and cell completely divide into two daughter cell. And when mitotic cell division occur four phase, you can see the structure prophase, metaphase, anaphase, telophase. Prophase is the first stage of mitosis where the cell chromosome of the cell divide into two chromatids and held together by unique DNA region is called centromere. And the prophase progress chromatid becomes shorter and thicker. And prophase includes cell division and centrioles and move to the toward the opposite pole. And metaphase is the second or longest stage of cell division where the chromatids lined up into the metaphase plate and chromatids are shorter and thicker still held together by the centromeres. You can see these are the structure of prophase. The calls and chromosome are arranged in the and in the middle prophase. Prophase is divided into three type: early prophase, middle prophase, and late prophase. In metaphase, chromosome align in the metaphase plate, and the centrum ancestral bind to the chromosome. And anaphase is the next stage of mitosis involved the splitting of each chromosome and into sister chromatid to form daughter chromosome. After splitting, chromatid move toward the pole due to sorting of microtubule. You can see these are the structure of the anaphase. Early anaphase is splitting of chromatids. You can see these are the chromatid and these are separated. And this chromatid move to the late anaphase. You can see this chromatid is moved to the pole and here the and telophase is the final stage of mitosis which involves the recognition of two nuclei and the entry of the cell into the next phase. You can see these are the structure of the telophase. Telophase is the next phase or last phase of the metaphase. You can see these are the structure where the two nuclei are nuclear membrane are formed and the chromosome completely go to the pole and here the cell are starts cytoplasm or divide constraint. During this phase, telophase, nuclear envelope is formed around the chromosome and form the two distinct daughter nuclei. Telophase indicate the meta M phase and which initiates Telophase indicate the M phase and which initiate the division of cell organelles and separation of cytoplasm into two cells by cytokinesis. Telophase is the end phase of M phase or mitotic phase and it further when initiates with the cell organelle separation by cytoplasm or cytokinesis. You can again see the structure of cytokinesis where the end phase of the telophase is the end phase of the metaphase. Here the cytokinesis phase occur and here is separate of completely formed daughter cell are separated. Next come to the cytokinesis. Cytokinesis is hollow movement of the cell. Cytokinesis is hollow movement of the cell and division of protoplast into the daughter cell, and a division of karyokinesis so that each daughter cell move to the its own nucleus, and cell organelle and mitochondria plastid Golgi bodies are also distributed in between the two daughter cell, and mitochondria and plastid undergo division of cleavage and detail of the known as a cytokinesis or followed by karyokinesis. Not follow cytokinesis does not follow karyokinesis. You can see the difference of the cytokinesis and karyokinesis and cytokinesis. First of all, karyokinesis division of nucleus into two daughter nuclei, whereas cytokinesis is division of cytoplasm into two daughter cells. Karyokinesis division of nucleus into two daughter nuclei, and cyto means division of cytoplasm into two daughter cells. Second one is a deorganization of nuclear envelope and appear that already replicate chromosome and splitting of equitable distribution. And in karyokinesis, nuclear is envelope is appearance and already replicate during the splitting of equitable distribution. In cytokinesis, no such elaborate mechanism occur and equitable distribution of the cell. Karyokinesis is the first step of M phase or metaphase in, and cytokinesis is the last step of metaphase. So this all about the cytokinesis. Here is the structure of the how chromosome enter. First of all, G phase, you can see these are the fiber thread like chromosome in G phase, where all the synthesis important material are synthesized, and you can see these are the S phase, where the chromosome start to like thread like structure, and it appear. In G2 phase, chromosome is formed the coiling structure, and in prophase, chromosome is completely formed and attached to the one pole. In metaphase, chromosome are arranged in the cell metaplate or cell plate, metaplates of the cell. In anaphase, all chromosome are daughter chromosome are separated and split into the chromosome is split into the daughter chromosome. You can see these are the chromosome is split into daughter chromosome in anaphase, and then again this forms cytokinesis, where where the daughter cell are formed, and then it also again start the G1 phase and S phase. This cycle are repeated when it required for division. So this all about the cell cycle and cell division. I hope you understand well. If you like this session, please like, share and subscribe. Thank you for watching.